if you're going out on Redfern train system, take the time to go past Cicada Innovations, where Dr. Alan Taylor's company is located. Back in the day, this was a disused rail station. You've seen the boilers out front, and today it's repurposed for collaboration and for talks on innovation and such. Dr. Alan Taylor is from the University of Sydney, the University Medalist in Applied Science, PhD at Garvan Institute across town, DNA House of Sydney. Alan's working with Clarity Pharmaceuticals as the executive chairman, developing radiopharmaceuticals for the treatment of cancer and other diseases. Please welcome Alan Taylor.
Thanks, Ashley. Hi, everybody. Just to make a general comment at the beginning, I spent a lot of time at conferences, presenting, and also just attending as attendance, and I've never seen this conference is incredible in the diversity of people who are presenting, everything from government, industry, all the way through. So I'm just a simple scientist. So and some would say sometimes an evangelical scientist. And I must say that last presentation, to have a banker talk about evolution, transhumanism, and the way society is living is fantastic. So well done. Okay? So to kick off, so our little goal at Clarity is to develop better cancer treatments. And that's to treat adults and kids in a safer and more effective way. But what I wanted to do, because this isn't specifically an oncology conference, I didn't want to discuss exactly what we do in detail, but I wanted to discuss it more in the relevance of IoT, given it's an IoT conference. But I have a confession. I've known Eitan for a long time, and he's asked me to present at this over the last few years. And when he first asked me, I jumped at the chance. I said, yes, I'd love to. It's nice and close to home. Makes it nice and easy for me, and it's great. And under my breath, I said, but what the heck is IoT? And I thought it was just me, but I come from an industry that maybe hasn't well recognized it. So I did what all good scientists did. I went to that fantastic research tool called Google, and I found a whole bunch of things that it potentially is, or certainly descriptions. So I did what all humans do. I chose my favorite, and I focused on that, and really started to think about more why isn't it relevant more in, say, clinical development than what it is today. So that led me on a path to get back to where does it actually fit in my schema of understanding as a scientist, and certainly as a scientist from where I focus on the translation of science through products and services to make the world a better place. So coming back to first principles and understanding exactly what science is, because science is thrown around a little bit like a political football, but when you break it down, it's really more about observing and measuring the world around us so we can make useful predictions. And then on the back of that, and where I more fit, as opposed to being a basic scientist, is more using engineering techniques to produce products and services, but always fundamentally based on scientific principles. And we always use, as scientists or translational scientists or clinicians or whatever, technology. And the technology that we use specifically at Clarity, for instance, are quite big imaging equipment, sequencing equipment, and trying to get an understanding of how cancer works, evolves, and more importantly, how we kill it. And this fundamentally, and this in particular, is where the internet works, and certainly the Internet of Things. It is a tool to enhance our ability to do science and engineering. And that science and engineering can either enhance the way the internet works, or better still, we can produce better products and services so we can increase the efficacy of cancer treatment. And fundamentally, this is underpinned by mathematics. And I know a lot of you probably find mathematics quite boring. I'm a bit of a nerd on it, so I'm going to share in more of a fun way why mathematics and where does it fit. And I've thrown up there a quote by Daniel Kahneman. Some of you might know him. He's a Nobel Prize winner and wrote a book, Thinking Fast and Slow. And really he starts to question the mind of humans, because we're not really designed to do mathematical equations all the time. And evolution has it that we simplify things, we have bias, we jump to conclusions, and all those sorts of things because we're processing so much information. And this has been over however long, talking about evolution. This has got us to the point we are because we are somewhat irrational, but that sometimes helps us in progressing our way of life. But about 300 years ago, we started to generate a little bit of data, not just think about things, but start generating data. And we started to really develop the scientific method. And the scientific method itself is underpinned by something called the null hypothesis, where we've chosen out 95% confidence level to test everything that we do. So if it's over 95% chance that something's going to happen, we call it significant. If it's under 95% chance, it's not significant, and we throw it in the bin, basically. And we don't publish results that are actually under 95% confidence. But where this is somewhat insufficient is we try and control all factors here so we can measure cause and effect. But in the real world, it's really different because those factors live in the real world, so it's hard to balance that out. But the null hypothesis isn't necessarily made to actually process this level of information. Now, this, for those mathematicians in here, probably know the difference between a null hypothesis. The one on the end there is then a Bayesian measure where we're actually gathering lots and lots of information and trying to work out probabilities. Okay, this is quite even though I've made it a fun slide, quite significant in the development of certainly pharmaceuticals over time, because at the moment, the way we delve out medicine is that we have a human at the front end with a patient, subject to bias. We have the null hypothesis that works through the FDA regulatory process, and we're not utilising at this point in time a lot of Bayesian methods to substantiate whether products work in society. And this here is actually the opportunity for IoT, because IoT is able to generate more information in society just by having everything connected, and we're able to generate data which is highly relevant. This becomes more significant in things like the treatment of children's cancer, where the numbers aren't great globally, so running phase 3 clinical trials are incredibly difficult. So we have to look at other mathematical ways to deal with it, and a Bayesian mathematical way is much more relevant. So based on this information, and I sort of touched on the point of where modern medicine fits, there's a number of things which are wrong with this picture, yet this is how we promote a doctor. He looks very smart. He's looking at an image, an X-ray. The issue here is, and we know that he's probably got an issue with his eyes. He's wearing glasses. We don't know whether his optometrist was necessarily the best or worst, whether he's up to date on his prescription. Yet he's looking at an image and using judgment to determine whether a patient is sick or not. Now, this in itself raises a lot of issues in the way that we process that information, given these humans subject to bias. We also have the situation currently where we run a clinical trial program where we get a large group of population or a group of people with a particular disease, and we treat them the same. And we don't necessarily build on the data to select those patients which are likely to actually have an efficacious effect. And when we treat that patient population and work it through phase 3 clinical trials, even though it might be proficient in a particular group of people, we're using more of a broad spray. And so that's when, even when we're treating people with things like chemotherapy at the moment, we go, let's try this, let's see if it works. The issue with this, especially in cancer, is that we're basically firing in the dark, trying to kill cancer. Because cancer, and to take a little bit of a delve into what is cancer, cancer is a separate organism that lives in our bodies that wants to basically kill us as a host. It won't die. It becomes immortal. We use actually these cells in research because we know they grow fast and don't die. They're fantastic in that sort of thing, but they're no good in our body. And they look at ways, they change blood supply, they change energy to them. They actually become invisible to the immune system. So using an approach which is rather archaic, where we're focusing more on the bias of the human mind and maybe a process of regulation which is still working on a null hypothesis, isn't enough to get rid of it, and it's not to date. So we're working towards the design of far more sophisticated drugs. We've got Keytruda, which some of you may have heard about, which is a fantastic drug that suddenly makes cancer, stops cancer from being invisible to the immune system, so the immune system can kill it. We've got targeted therapies, and that's the area that we're in, peptides and antibodies that specifically target the cancer. And we've got a whole bunch of other types of therapies, but these are incredibly complex. And not only that, they're incredibly expensive. Not only the development of them, but obviously then when you're producing them, because they're more biological, they cost a lot more than a small molecule. And this is a bit of an issue. So to better treat our patient population, we probably have to better select who is going to be a responder and who's not. So this here, in the diagnosis of cancer at this point in time, there's a number of ways that we work through a process, and some of those are genetic testing to determine potentially what type of cancer it is. We use imaging technologies which are evolving and getting better to the point where we can quantify what's going on. And we use histopathology. So this happens in a path lab to determine exactly what type of cancer it is. Now this is fairly siloed at this point in time. And I just want to focus on the first two because histopathology is well characterised, but the next two are evolving over time. And there's a lot of publicity around things like genetic testing. And for instance, a place that I was at previously, the Garvan has gone very, very much focused on genetic testing as a way, and it's fantastic, and it's going to lead to some significant milestones in the future. But what happens in the genetics of cancer is interesting. So in the situation here, this is what a theoretical model is. You get a couple of mutations and then suddenly it turns into cancer. And you can see there that it's rather homogeneous. It's one type of cancer and expressing the same genes, etc. But the real world, once again, is very different. And it makes it very hard in that not only, for instance, pancreatic cancer, not only is it different between person and person, it's even different within that one person. And it might be different in the metastases, so when it moves and aggressively grows, the cancer might be different between the metastases and the primary. And even in the one cancer, it might be different. So when you take a gene array, you don't know exactly what's going on, what clone you're actually picking up on. And where science is going now, we're starting to find things like when we dose with chemotherapy. See here we've got clone 1 dominance. So clone 1 could actually be dominating clone 2. But clone 1 might be sensitive to chemotherapy and clone 2 is not. So when we kill clone 1, clone 2 may actually grow out of control. And this is, we're actually focusing now, chemoresistance might actually be that. So we're starting to better understand how it works. But genetic testing will only get us so far. So just here, I've put up here, and the comment underneath, the imaging is more, it is a lot more than just an image in itself. It is significant data. And now we can quantify. So now it's time to move forward with more imaging as well to better characterise what's going on. And we can understand exactly how much drug, for instance, is hitting a tumour, how much is going through rate-limiting organs, and then how much to dose. And we're still working through that process, but it's going to take a lot more coming together of knowledge to make sure we can progress this through FDA regulatory processes. But the sensitivity now of imaging is getting better and better, and the quantum mechanics lecture earlier, even looking to progress this even further, so we can better characterise what's going on at that cancer level. These are our own images, and these are the images actually over time of a drug hitting a large pancreatic-based cancer. And the red part is how much is there on the cancer. So we know it gets there really quickly. We know it stays there over time. And we know how much is going through the kidneys, which are the green parts on the outside. And now we look to, we got so much information out of that, which the data can feed straight into a process of them being more predictive on how we treat with a particular therapy. And really the level of detail we can get out of these images now is amazing. And as I said, even getting better. So where does IoT fit then? And really, when we think about IoT, and certainly the meaning that I chose at the beginning, who came out of Cisco, trying to bring these different devices together, the devices I'm talking about to actually build out this data is gene sequencing, understanding the limitations of gene sequencing, but also the benefits. And as we get to better understand the genome and predictability, bringing that data into a central process of better understanding the cancer. Detailed patient history, feeding straight into a database to better understand what's going on. Imaging. As I said, it's data. It's actually just not a picture or an image. It is actually detailed data, exactly what's going on in the process of drug delivery. And then histopathology, and also all of the published data in relation to cancer treatment that's been published since we started publishing research. This way of processing allows us to get away from what is the old way of actually treating a whole patient population and hoping for the best, to being more specific, more targeted, and I can use a number of words like personalised and those sorts of things, working through that process so we become more efficacious to the patient groups that we want to actually achieve an outcome in. So I want to finish this off, and because there's actually, this sort of discussion was more to open up ideas and opportunities, because I'm not necessarily an IoT person per se, but there's such a significant requirement of IoT in what I do, and that is to increase efficacy in cancer treatments. So Clarity itself, we have an ability to diagnose in the first instance, select patients, and then hopefully work towards a better treatment. But what we're also able to do, we know how much dose is getting there to the tumour. We know the rate limiting organs, so then we can optimise dose to maximise efficacy and minimise side effects with a therapy isotope. It just so happens that physics has delivered us copper-64, which is ideal for imaging, and copper-67, which is ideal for therapy. Now, where this resonates for us at Clarity, in particular, is in the treatment of children's cancer. Neuroblastoma is one of the biggest killers of kids under 5. At the moment, we're using high-dose chemotherapy to treat these kids. And high-dose chemotherapy, these are drugs that have been developed somewhat, even up to 80 years ago, and they have significant side effects. But unfortunately, it's the best we've got at the moment. So we know we can hit the target in neuroblastoma, and we know that we can be efficacious with copper-67. The issue that we have at the moment is that we need to work and build greater data sets to understand the link between diagnosis and therapy. Professor Rod Hicks from the Peter MacCallum Cancer Centre said in a paper published only a couple of years ago, the difference between us and magicians is magicians know what they're doing. And so from our perspective, what we're trying to do is guide treatment, better understand the cancer, and optimise efficacy. This doesn't happen, and once again, to draw on a previous presentation, collaboration is essential when you're actually progressing these sorts of things. And so we collaborate with people not only here in Australia, some of the best institutes and hospitals in Australia, but it's incredibly important to also collaborate with those internationally with significant experience in developing these sorts of opportunities.